Hi, good day, everyone. Welcome back to class. It is learning time today again. So I'm welcoming every one of us back to class. To my returning subscribers, thank you so very much. And if you are coming across this channel for the very first time, this is where we learn biochemistry in simple English. Do stick around and enjoy the ride with us. Also, please subscribe and share this video for other people also to enjoy this video and learn with us. Now, from our previous class, we discussed extensively on amino acids. Now, today we want to discuss about protein, but there is no how we will discuss protein without discussing amino acids. Amino acid, as we stated earlier, that they are organic compounds that have this basic structure. They are also the building blocks of proteins, meaning that, let's take for example, this is one amino acid, this is another amino acid, this is another amino acid. So if I join this amino acid A to amino acid B to amino acid C, this will now give me a particular protein. So this, now if I'm holding it together like this, I have a protein. But if I want to break it down, I will say that in this particular protein, I have 3 amino acids. When amino acid A join with amino acid B, for it to stand like this, there is a bond there, and that bond is what we call peptide bond. Now, when this one wants to join with this also, we have another bond there, that is peptide bond. Now, how do we get this particular peptide bond? Take for example, we have one amino acid here, we have another amino acid here. We now want to join them together. The reaction will occur between the carboxyl group of one amino acid and the amino group of another amino acid. This particular carboxyl group will be activated by ATP. When ATP activates this amino group will now attack the carboxyl group, the activated carboxyl group. When this happens, dehydration occurs, meaning that the OH here and one hydrogen here, one H here and OH here will join together, which will give us H2O. Water will be released. So if we, after removing this OH and one hydrogen from air, what we are left with is CO, then NH. This is what we refer to as peptide bond. So this is the bond that we hold this amino acid A and amino acid B together. So if I now want to add another amino acid now, which is amino acid C, so between the carboxyl group of this particular amino acid and the amino group of this particular amino acid, when they join together, there will be release of water and there will be formation of peptide bond. And that's how this peptide. So with this now, you know, we have, this is amino acid 1, this is amino acid 2, plus another amino acid 3, plus amino acid 4, plus amino acid 5, etc. So here we'll now be having the peptide bond here, peptide bond here, peptide bond, peptide bond, peptide bond, like that. So this is what will glue all these amino acids together to give us a particular protein. Now, a particular protein could contain thousands of amino acids, and they contain 50 amino acids. It depends on the particular protein of interest. The most important thing is that the composition of this amino acid sequence now, how these amino acids are joined together, will now determine the structure of the proteins that we have. You know, amino acids are building blocks of protein. So it will determine the structure of the proteins that we have. Now, the structure of a particular protein will now determine the function in which this protein will perform. Now, what are proteins? Protein is just the functional molecules of life, meaning that protein is the basis of everything in our body. So can you see that? Can you see how amino acid is important now? So that is why in biochemistry, when you see us say anything, cannot stop talking about amino acids and protein. Because biochemistry on its own is the study of every chemical reaction that is happening in our system. What are the functions of these proteins? Number one, they serve as enzymes. They help in catalyzing reactions. Number two, they also serve as structural components. Structural components of the body, of different cells, and the rest. Number three, proteins help in transportation. Example, hemoglobin. It helps in carrying oxygen through the blood and circulates in the system. That is part of the function of a protein. Another thing is that proteins help in signaling. In example like hormones that send messages, okay, this you need to stop, this you need to start, that those are works of protein. Also, receptors like a key to the body. Okay, did I recognize you? Okay, I think I can recognize you. Then the receptor will open the door for a particular molecule to enter. And in case the receptor did not recognize the key that is coming, it will not open. So these processes are being carried out by protein. Can you say why protein is very important as a biochemist? Let's check different structures of protein that we have. Basically, we have four protein structure. We have the primary structure, we have the secondary structure, we have the tertiary structure, and the quaternary structure. Let's take them one after the other. The primary structure of a protein is the linear sequence of amino acid. The linear sequence of amino acid. Let's say, from our previous example, let's say this is amino acid 1, which join to amino acid 2, which join to amino acid 3, join to amino acid 4, then 5, then 6 now, and like that. And we said that this bond is peptide bond, which is CONH. That is the peptide bond that joins this amino acid together. So now the linear sequence of this amino acid, that means the straight arrangement of this amino acid, that is what we refer to as the primary structure. The folding of this linear structure, which could be folded in form of a beta sheet or alpha helix, is what we refer to as the secondary structure of a protein. It's now being stabilized by hydrogen bond. Which is the hydrogen bond that will stabilize the folding of this linear shape that is now folding either into alpha helix or the beta shape. This, you can see that if there is any alteration in this primary structure, definitely it will affect the secondary structure, because we are getting our secondary structure from our primary structure. Also, in this secondary structure, the R group, which is the side chain here, we have a way of pointing outward. Now, let's move to the third one, which is the tertiary structure. The tertiary structure is the overall 3D structure of the protein. Combination of secondary structure. The secondary structure, when they come together, is the one that will give you the tertiary structure. And we also said this tertiary structure is the overall 3D shape. Overall 3D shape. So, can you see how the progression is going down? Quaternary structure now is the joining together, coming together of different tertiary structure, or we could say calling together of different polypeptide chains. And what do we mean by polypeptide chains? Polypeptide chain, as the name implies, poly, more than one now, then peptide, small fragments of proteins. So when these protein now join together, we have a polypeptide. So coming together of polypeptide chains is what will give us the quaternary structure of a protein. The tertiary structure, you remember in our secondary structure, we said that the bond that help us to hold them together is hydrogen bond. But for the tertiary structure, the bonds that help us to hold them in place, now you can see that they are becoming bulky. So the bonds that help us to hold them in place include one, disulfide bond, two, hydrogen bond, three, ionic interactions between the proteins, hydrophobic interactions between them will also help to hold them together. The next thing we'll be moving into now is protein sequencing, arrangement of amino acids that make up a particular protein. That's what we'll be discussing in our next class. See you in the next class. Bye bye.